Brad, come on up. So this, you might notice in the short talks for today and tomorrow, we've got some sort of non-R but bioinformatics related talks, and this is one such talk. So Brad won best prize at the Combine Student Symposium earlier in the week, talking about the work that he's going to present here today.
So sorry if you've heard it a few times before. This is a 3-minute version at least, so at least you don't have to sit through a longer version again. Cool. Okay, so I'm going to be talking about cluster significance analysis and single-cell RNA-seq data. So the package I've implemented this in is called Cytocipher, which is in Python. Okay, cool. So we heard from Aaron earlier the dark arts of taking a single cell Cowie matrix and converting into a UMAP with our tentative groups of cells. And so we also heard about the pre-processing steps in this, but I'm just going to focus on the clustering, the post-hoc differential expression, and determining that UMAP space. So ideally, when we do our clustering, we do a post-hoc differential expression, as we heard earlier, and you get marker genes for each one of your groups. And ideally, these genes align with each one of your clusters. So you can say this cluster is different from the others in that it expresses this gene or combination of genes. However, a lot of times this might not be the case because we've overclustered or underclustered, et cetera. And so we have to redo our clustering. So we're consistently changing our resolution or manually merging different clusters. So to try to solve this problem, I've developed Cytocipher. So the key idea and the key input of this is actually the genes by cells matrix, and we have a tentative group of labels for each one of our cells. We then get our marker genes for each one of the groups, and then we split these marker genes into positive and negative sets. So what makes the blue cluster different from the orange cluster is it doesn't express gene D, for instance. We then get a summary statistics for each individual cell for how well it matches those positive and negative indicators of belonging to each respective cluster. So we end up here with the ideal case when we have transcriptionally distinct cells, cell groups, is that the blue cells in the blue cluster score well for belonging in the blue cluster, and so on and so forth. So we get this matching along the diagonal here. However, when we don't have transcriptionally distinct populations of cells, we have this kind of cross scoring between different groups. And so if you just apply a statistical test on this and there's no statistical evidence that these are transcriptionally distinct populations, then you just merge them to create a new group. And so if you rescore, you should see transcriptionally distinct populations. So I've applied this in multiple different settings. So just on healthy tissue, on disease development, and also perform ground truth quantitative tests using the Tabular Sapiens Atlas as the ground truth, and artificially splitting known cell types into 3 different groups, and then seeing if it can merge correctly the non-significantly different groups within a cell type and keep the cell types separate, and that's performed really well. And each one of the biological examples, it's identified new subpopulations, or in the disease case, disease-relevant subpopulations. So this is currently implemented in Python, and it's interchangeable with the ScanPy ecosystem. So my plan is to talk to Aaron after this about using Basculus so that I can get this into R, so we can use it with single cell experiments so it can be more helpful to the broader bioinformatics community. So I just want to thank my supervisor, Mikkel, Stefan, and also Michael and different members of the Burden Lab. There's a preprint here, and there's a GitHub here. So if there's any questions, I'll be around. Cool. Thanks.
Thank you, Brad.